Good morning everyone. Today I will be presenting a JC on a topic, a novel circulating miRNA panel for non-invasive ovarian cancer diagnosis and prognosis. This article was published on 5th of August 2022 in British Journal of Cancer by Ghalawat et al. Now, ovarian cancer is a highly malignant cancer among all the gynecological cancers with a 5-year survival rate of 45%. And majority of the patients, they present with advanced stages of ovarian cancer, which is having a survival rate of less than 20%. According to Globocan 2020, worldwide incidence of ovarian cancer was found to be 1.6% and 5-year prevalence rate of 21.3 per 1 lakh of population. And according to ICMR, in India, incidence of ovarian cancer is between 5.4 to 8 per 1 lakh of population. Since more than 70% of all ovarian cancer patients are diagnosed at late stages with a poor prognosis, so it needs the development of screening biomarkers. This is an overview of the types of ovarian cancer. It has been classified into germ cell, sex cord, stromal and the surface epithelial tumors. And surface epithelial tumors are the most common type of tumors encountered. And diagnosis is made by standard guidelines. Currently, the existing biomarker for the screening of ovarian cancer is CA125, which is carbohydrate antigen 125, which is having a sensitivity of less than 60% in early stage ovarian cancer and up to 80% in advanced stages. CA125 is being measured in a lab in this machine, Siemens Advia Center XP, by the method chemiluminescence. These are the current emerging biomarkers in the ovarian cancer. Human epididymis protein 4, paired box gene 8, CDKN2A and microRNAs. Current paper focuses on microRNA and they are studied for their utility as a biomarker. Various types of studies have been conducted on individual miRNA in ovarian cancer. This is one of the study which was conducted by Oliveira et al., in which they have taken the plasma of 46 patients. They have investigated the plasma profile of 46 microRNAs, and in which they have taken 190 patients with malignant, total 190 patients with 95 malignant cases and 95 benign cases. And in this they found that 4 microRNA, this is microRNA 200C3P, microRNA 221-3P, microRNA 21-5P and microRNA 484 was found to be upregulated, and 2 of them, 195-5P and microRNA 451A, was downregulated. And among all these 6 candidate miRNA, microRNA 200C3P, it showed the highest predictive efficiency with area under the curve 0.78. Now coming to the microRNAs. MicroRNAs are non-coding RNAs of 20 to 25 nucleotides long. They regulate gene expression by repressing translation or mRNA degradation. They are transcribed in nucleus and exported to cytoplasm, resulting in the formation of mature microRNA. First miRNA, Lin4, was discovered in C. elegans by Ambrose et al. In 1993. Later in 2000, a detection of miRNA in humans was discovered. And with the coming years, in 2005, first microRNA transgenic mouse was created, followed by microRNA detection in plasma in 2008. And since then, thousands of microRNAs have been identified in the various conditions like cancer. Now coming to the biogenesis of microRNA. MicroRNA are formed in nucleus from DNA. First, pri-mRNA is formed, and with the help of enzyme DROSHA, it is a type 3 RNase enzyme which helps in the formation of pre-miRNA. And this pre-miRNA, then it is exported from nucleus to cytoplasm with the help of exportin 5. And there, pre-miRNA is being converted to microRNA duplex with the help of dicer. Dicer is another type 3 RNase enzyme. After that, this duplex is unwound. Mature RNA is incorporated into the protein complex. This protein complex is named as RISC, RNA-induced silencing complex, and it will guide this RSC to the target mRNA. This is the target mRNA. If it is found perfect complementarity, then it will degrade the mRNA and it will repress the translation later on. And if there is imperfect complementarity, then mRNA degradation will take place. And this is how mRNA regulates gene expression. The role of microRNAs in health and disease. Normally in healthy individuals, miRNA helps in regulation of multiple genes, whereas their altered expression can lead to pathogenesis of various diseases such as cancer. For example, the microRNAs have been broadly classified into the oncomes and the tumor suppressor microRNAs. Normally they keep a balance with each other, but in cancer conditions there is the disturbance of this balance, and which leads to the tumor growth, either oncomere overexpression or underexpression of tumor suppressor microRNAs. And these are the microRNAs which are classified under either as oncomere or tumor suppressor microRNAs. Now, how they come into the circulation, what is the origin mechanism of these microRNA to come into the circulation? Basically, extracellular RNA, which is outside the cells, it exists in 4 forms, either freely circulating, through protein complex, through bound with the protein complexes, bound with the lipoproteins, or active secretion via extracellular vesicles. In normal condition, if apoptosis of cell takes place, then miRNA will be released into the circulation. Now, coming to article of today's JC, which is a novel circulating miRNA panel for non-invasive ovarian cancer diagnosis and prognosis. Authors highlighted the potential of circulating free microRNA as no other biomarker is available till now. And currently CA125 is the only blood-based marker which is used for the screening of ovarian cancer, but it is very non-specific and less sensitive. The aim of this study was to highlight the potential of cell-free microRNA and microRNA panel for ovarian cancer diagnosis as well as prognosis. The main objectives of authors was to identify the prognostic role of circulating free microRNA for early stage ovarian cancer, to determine the diagnostic and prognostic role of 7 microRNA signature panel, and lastly to determine the association of these total circulating free miRNA with various clinico-pathological features of ovarian cancer. The study was a case control study. It was conducted at University Hospital and National Center for Tumor Diseases, Heidelberg, Germany. Total 244 study participants were enrolled in this study, and after screening, 99 healthy controls were taken, 100 ovarian cases in which 80 were serous adenocarcinoma and 20 were others. And in benign ovarian lesions, the patients were 45 patients were recruited. Among those, mucinous cyst adenoma, serous cyst adenoma, ovarian cyst, fibroma, mature teratoma, and other categories were included in these benign lesions. After the proper consent and the approvement by Ethical Committee of Heidelberg, Germany, blood was collected in EDTA vial, and later on miRNA expression was done through RT-PCR, and CA125 was analyzed through ELISA in this study. Before going on to what they did, this is the general procedure we follow in our labs. So after collecting body fluids, it can be a serum, blood serum plasma, CSF, urine. From all these body fluids, we can isolate microRNA. So microRNA isolation is done from phenol chloroform method, and to check the quality of microRNA isolated, we check it through nanodrop instrument, and later on we quantify microRNA through RT-PCR or microarray or NGS, and later we can see the expression and analyze it for diagnosis and prognosis. The sample was collected in EDTA tubes. 3 tubes were taken, 3 ml blood in each tube. They have performed the replicates of this. And after centrifugation, the samples were stored at -80°C. And later on, circulating microRNA, they were isolated using nucleospin microRNA plasma kit according to the manufacturer protocol. Later on, they are quantified using qubit microRNA assay kit and the qubit fluorometer 3.0. And RT-PCR was done on QTAR instrument. These were followed according to the manufacturer protocol. This is how further they went about. They checked since the total circulating free microRNA levels were associated with adverse clinico-pathological features of ovarian cancer, and they had also significant prognostic impact on survival of ovarian for overall survival in ovarian cancer. So author hypothesized that it can have a diagnostic potential too, but there was no significant diagnostic potential was found in case of total circulating free microRNA levels. So they created a 7 microRNA panel for diagnosis of ovarian cancer. So this is how the 7 microRNA signature panel was selected. The authors have downloaded the NGS data, and using that NGS data, they identified signature of circulating free microRNA. So in this, only 192 candidate miRNAs were eligible for analysis. Later on, top 20% differentially expressed microRNAs were taken and cross-checked with Analyse et al. Study. So finally, the 9 microRNA signature was formed, and to investigate the predictive value of this 9 circulating free microRNA, they measured it by RT-PCR and performed the multi-lomian logistic regression analysis, and finally the 7 microRNA signature panel was formed and it was validated with Analyse et al. Study. And these are the 7 microRNA panel. MicroRNA 92A, 200C, 320B, 320C, 335, 375 and the 486. These were the statistical methods which were used in this study: Kaplan-Meier survival plots, Cox proportional hazards regression model, and receiver operating characteristics curves. Now coming to the results. After statistical analysis, this table is showing clinico-pathological characteristics according to the circulating free microRNA levels. So to assess whether the increased levels of this total circulating free microRNA are associating with the clinical features of ovarian cancer, so the authors have dichotomized patients into high or low circulating free microRNA groups according to the median. Later on, they also dichotomized CA125 according to the median into high and low circulating free microRNA, taking median as 35. Less than 35 international unit per ml is low circulating free microRNA group, and more than 35 is high circulating free microRNA group. And authors have found that higher levels of circulating free microRNA were significantly associated with CA125, presence of distant metastasis, and mortality. So it came to the conclusion that higher circulating free microRNA levels correlated with adverse clinical features of ovarian cancer. Now to see the prognostic role of circulating free microRNA, authors have carried out Kaplan-Meier survival analysis. So these are the Kaplan-Meier curves. On x-axis, years elapsed are there, and follow-up time period for this study was 3.8 years with a median of 1.8 years. And on y-axis, overall survival is taken. So from Kaplan-Meier curve, they compared overall survival. So higher level of baseline circulating free, this high level of baseline circulating free microRNA were significantly associated with overall survival, as p-value is less than 0.001. Since majority of the patients present in advanced stages. So authors have included only stage 1 to 3 in figure B, and they have found similar results, indicating that circulating microRNAs can be an independent prognostic marker in ovarian cancer. And to validate these findings, multivariate Cox regression analysis was done. So in line with the Kaplan-Meier analysis, circulating free microRNA levels were highly significant for overall survival in this multivariate analysis, as hazard ratio is 1.83 and p-value 0.001, confirming that the total circulating free microRNA is an independent prognostic marker for the overall survival in ovarian cancer. These are the ROC curves. In figure A, they have taken their own cohort, that is Heidelberg cohort, and in B, they have validated their results with the external cohort, that is by Elias et al. So it was observed that 7 microRNA panel could distinguish control from cases with an area under the curve 0.87, and it could distinguish control from the benign cases with area under the curve 0.77. Similar results were found in this external cohort also, and they are having a good discrimination potential. However, the cancer determination was better in case of cases versus control due to the high area under the curve. Now authors wanted to assess diagnostic potential of circulating free microRNA panel, specifically in early stages of ovarian cancer. 7 microRNA signature panel could detect early cases from control with an area under the curve 0.81 and late cases versus control with area under the curve 0.91. Similar results were found in external cohort, and they have validated with this external cohort. So it was a good discrimination potential found in both the cohorts. However, late stages cases determination was better due to high area under the curve. Now authors have combined this panel with the current existing CA125 marker for the ovarian cancer diagnosis. So in figure A, they have taken benign cases, they have compared the benign cases versus control and cases versus control, and in figure B, they have specified early and late cases versus control. So they analyzed that the 7 microRNA panel along with CA125, it could distinguish cases from controls with area under the curve 0.97, which was retained with an area under the curve for early stages versus control, that is 0.93. It was a better result. Now coming to the discussion. Authors found significant association of total circulating free microRNA levels with adverse clinical features in ovarian cancer, and total circulating free microRNA levels could be independent prognostic marker for survival in ovarian cancer, and 7 microRNA panel in combination with the current screening marker, that is CA125, could be diagnostic for early stage ovarian cancer detection, and it can be easily applied in clinical setting, and adding it with CA125 will remarkably improve the testing. So these are the various studies which were conducted by Meng et al, and they reported significantly higher levels of this individual microRNA 200C, and Cirillo et al has also investigated expression of microRNA 141. It is also a member of microRNA 200 family, in combination with CA125 and HE4 for ovarian cancer diagnosis. And these studies were corroborating with this present study. Similarly, Oliveira et al has distinguished benign from the malignant tumors with overall diagnostic accuracy of area under the curve 0.96 when using the 2 microRNA in combination with CA125. And Cance et al, they found members of this microRNA 200 family to be upregulated in ovarian cancer patients as compared to the healthy controls. Authors had shown levels of circulating free microRNA as an independent prognostic marker in breast cancer also, and it was correlating with the unfavorable tumor features as they have than in ovarian cancer too. So further studies are required so that it could be used as an add-on marker for early diagnosis of ovarian cancer. This study concludes that total circulating free microRNA levels could be independent prognostic marker for survival in ovarian cancer, and if this panel, in addition to current existing marker, 7 microRNA panel, in addition to current existing marker CA125, may enhance the diagnostic ability of ovarian cancer. Limitations of the study: they relied on the miRNA profiling data from the another study, and external data served as a validation cohort in their study. So we can't be sure that selected miRNA are most representative of their cohort or not. And panel signature was optimized to distinguish cases from healthy controls rather than from benign cases. Patient had a relatively low follow-up time, which have hindered the prognostic studies. Imbalanced cancer cohort with mostly late-stage serous adenocarcinoma patients, and sample size calculation was not provided. In this, some of the limitations authors have included in their paper, and inclusion criteria and the exclusion was not mentioned properly. It was a single center study, so couldn't collect most diverse group of patients, as it reduces the variability. Incomplete results of miRNA expression, and it was not mentioned clearly whether this panel, 7 microRNA panel, could predict prognosis of ovarian cancer or not. This is a strobe list. Title and abstract, yes. Introduction, yes. Background rationale, yes. Objectives, yes. Methods given, yes. Study design, yes. Settings, yes. Participants, yes. Variables, yes. And data sources not available. Bias not available. Study size was quantitative variables, yes. Statistical methods, yes. Results, yes. Discussion, yes. Limitations, yes. And funding also. Thank you. And